We are ready to put on our big boy and big girl pants. And the reason that I say that is because in this lab experiment that you're getting ready to do, or maybe that you are in the process of doing now, is actually another DNA lesson. But this time, it's actually using your own personal DNA for a change. Okay, so leading up to this moment, we probably have given you these kits, and these kits have had, I'm not going to say fake DNA samples, but these kits have already had DNA that has been extracted that has saved you a little bit of time. So we have injected these samples into gel electrophoresis, and then we have kind of compared these lanes in gel electrophoresis with each other. So at this moment, you probably have done something that is more like a parental test. So who is the parents of this child? And we know that the child has half the father, half the mother. So we can play match game to figure out where half of the child's DNA is coming from. So that way we've identified the father of the child. The other lab experiment that you probably have done at this point is a crime scene. That doesn't even look like the word crime. What on earth am I doing anymore, right? I can't even write the proper way, much less talk the proper way. So let's try this again. Crime. There we go. That's a little bit better. All right. So the other lab experiment that we've done is a crime scene investigation where this is going to be a perfect match. So what we were looking for was a suspect or one of the suspects perfectly matching the DNA sample that was obtained from a crime scene. And we know that maybe it puts them at the presence of the crime scene itself. That doesn't mean that they've done the crime, but it at least puts them a little bit closer to the crime or the suspect or the victim or whatever the case is. So in this particular video, we're actually going to be focused on our own personal DNA. And each one of these cases was something similar, right? And the reason that we do this is because we want you to go through some of those training protocols a little bit before we actually start using real things, doing the real experimentation part of this DNA extraction, the PCR, the electrophoresis, and so forth. Get your feet wet. So that way, when you get to this point, you'll feel a little bit more comfortable doing what you're doing. So for this particular lab experiment, what you're going to find out is that we are getting ready to undergo the process of number one, extraction. All right. And this is normally what you're going to do on the very first day of lab with us. You're going to go into the lab. You're going to probably swish, and you're going to swish your mouth with some saline solution, which is just salt. And that will help get your cheek cells all nice and bursted open. So that's kind of what we're after, because that's where the DNA is going to be held. Now, we can do cheek cells, but this process can also happen with hair. This process can also happen with skin. That is not a big problem at all, but we just need a source to get your personal DNA extracted from. And that's what the first part of this lab experiment is going to do. So for this reason, number one, we are using real, ding ding, real DNA samples for a change, when the other two, not so much. I mean, at least not in the perspective of a real DNA sample coming from someone. So in this experiment, we're actually going to be extracting real DNA, handling real DNA for a change, but just to make it a little bit safer in the laboratory, we're going to allow you to analyze your DNA that you have gotten from your cheek cells with your whole process of swishing your mouth in with our salt solutions, spinning them down, centrifuging them, bursting your cells open, and then eventually going into the PCR step. The second part of this lab experiment is a real PCR finally. So what you're seeing with the parent and with the crime scene, a lot of times they've mentioned PCR in those lab experiments, and they've referred to the PCR process. In some cases, they've went into more detail than maybe the other one did. And that's okay, but they're at least saying, hey, remember, PCR is actually out there and it's getting done. Well, in this particular experiment, you're going to undergo that real process here with an actual thermocycler, adding your DNA base pairs, everything that's needed for the primers and the replication process and so forth. We're going to go through that replication process for real. for the first time this time. And then we are going to amplify actually our own DNA sample, right? And this is, again, the first time that this is going to be done with us. So that's even better. That allows us to kind of get a little bit more real, actual hands-on lab experience using these processes in a laboratory. And then finally, number 3, we're still undergoing the electrophoresis of the PCR amplification, extraction kind of process. And this is something that you have already done really in the other 2 labs that we've already completed. There's going to be no difference really here. The electrophoresis is still going to include an agarose gel. It's still going to include a buffer. You're still going to inject your samples into the gel and allow them to band out, and then analyze the banding that you begin to see from your staining and destaining process. So this piece in particular is like, this is like nothing new. We've already seen it. We've already done it twice. But for a change here in step number 1 and step number 2, the extraction is going to be somewhat of a newer process for us. And the actual PCR, of course, is going to be a newer process for us as well. All right, so what we're actually doing here in this experiment, you're going to get this nice little handout that's going to come along. This will include your laboratory directions and your lab procedure that we need to follow, the steps that are going to be involved. You've seen things like this before from us already, and this experiment is not going to be any different. All right, but there are maybe some new terms that I want to focus on here, and these new terms are going to be a couple of different abbreviations that I want you to pay attention to. So first off, we've already talked about it really before, but your DNA is actually going to come to us in the package of what we call 23 pairs of chromosomes. And these chromosomes are going to be important because that's where all of your genetic information is going to be in. So I want you to think of this DNA strand, this big long strand of DNA with all these base pairs. They are packaged together and they are organized in a certain form or certain fashion. We're going to call this chromosome in a way, and those chromosomes are going to house this genetic information that we want to know more information about. So there are times that we get this something that we call a polymorphism. So a polymorphism is just a slight change. That's all that is. So for instance, your DNA strand might have a certain number of base pairs in the order that they show up in, and then a polymorphism is this idea that that particular DNA, one of those little bitty base pairs can be substituted for another little bitty base pair, or something similar like that, right? That is what we call a polymorphism. So there's a couple of different polymorphisms that show up in a DNA strand, and this is really what we start comparing people's DNA against each other with. So your DNA is made up in a certain way with a certain order of base pairs. My DNA is made up of a certain way with a certain order of base pairs. And what we're going to find and discover is that the order of those base pairs are pretty important because those create these changes in the DNA strand, and those changes can be compared in DNA PCR electrophoresis, as well as many other things. So there's going to be a couple of different terms for these polymorphisms. So the very first one that we're going to look at is called the SNP. That's what I refer to it as, SNP. And an SNP is going to stand for a single nucleotide polymorphism. So a single nucleotide polymorphism is just that. So single means one, nucleotide is referring to the base pairs. So this is one base pair that has been switched out that's different from mine than from yours. So that is a polymorphism. It is a change, but it's a change in one single nucleotide, aka base pair, and we call that SNP, which is going to be SNP abbreviation. All right, pretty important. Next up, maybe that one little base pair, it's a little bit more than that, and that's where we get this term called a short repetitive stretch. And that short repetitive stretch is going to be called a short tandem repeat by a formal definition. And short tandem repeats are abbreviated as STRs. And what you need to think of this as, well, it says short, so these are not very many base pairs at all. Repetitive means over and over and over and over and over. That's what we're going to see. And this really brief section of DNA that repeats over and over and over and over and over is a short tandem repeat, or STR. So I'm going to have certain STRs, and you're going to have certain STRs because our DNA is going to be a little bit different. And this is more than just one base pair that's a switch. This is going to be a grouping of base pairs that show up repetitively over and over and over. And then if they're not brief or short, then what we're going to find is another version of polymorphism that is called a variable number of tandem repeats, and that is a VNTR. So these are often a little bit longer, but they also are repetitive segments that are going to show up in mine, but maybe not in yours. So these three abbreviations are pretty important because they're often referred to over and over and over. We have our SNPs, or our SNPs, which is just snip, snip, right? Just a little bit. That's a change, one base pair. Then we have a grouping of smaller groups of base pairs that are repetitive, and then we also have a variable number, which is a little bit longer, but they're also repetitive, that begin to change. So these are different types of polymorphisms that we can see in our DNA strand. Now, most of these are what we call non-coding regions of DNA. They will often happen. If they do happen, it's not going to be drastic. It's not going to cause any kind of genetic deformities or anything like that. These are what we call non-coding, which means in the huge, big, long DNA strand that I have in front of me, these polymorphisms happen in areas that really aren't drastically important, that really don't mean anything. So non-coding, that's what that's going to mean. However, sometimes they do happen in the coding region of DNA. This is just a polymorphism. It just randomly shows up because we're going to find out that the non-coding region is actually the majority of the DNA. That's where most of it is actually going to be, and just a really tiny percentage of DNA is actually what we would call codes for something. So that DNA retains a message. That message is going to, associate with RNA or protein or whatever the case is, to give us just genetic information. So if those polymorphisms happen in a coding area of DNA, what you'll find out is that it will disrupt a gene, and it can cause a specific disease, or a genetic kind of mutation, if you want to think of it that way. So with that said, that's really what we're focused in on this particular lab experiment. And now you maybe are a little bit more familiar with these three abbreviations of SNP and STR and VNTRs. So now we can begin this discussion of what we call a DNA fingerprint, right? And a DNA fingerprint is starting to look at these polymorphisms. In whatever form that they come into, these polymorphisms can be compared from person to person or sample to sample. So this is what we're looking at eventually when we get to the electrophoresis. That is really what you've looked at in the electrophoresis gels and the other prior DNA experiments that we've done, even though they were what I would call these pre-packaged assembly kits. But this is kind of what we're looking for, right? Because your DNA is different than mine, and we need to figure out how is it different than mine. And if we can show that up on the electrophoresis, then we can start identifying and playing match game between the DNA samples that we're going to find. And it's these polymorphisms that allow us to see the different banding and the different ways that the DNA sample shows up on the electrophoresis from sample to sample. So that is why polymorphisms are so important, because it's going to help us determine what we see on the electrophoresis gels. And that's what you're seeing over here to the right hand side of the screen. So here, if let's say that we were on a crime scene, as long as we've got like a hair sample or some type of blood sample or some type of skin cell or just a cell itself, but skin cells, cheek cells, and so forth, we can take those samples and we can burst them open to release the DNA on the inside. And with that said, you can also take direct blood, right? No problem there. This DNA gets released. This DNA has to be cleaned up a little bit because there's tons of other things inside of cells that we don't necessarily need. It's just like trashy, messy rooms, and we just need to get the DNA out and get it as clean as possible. So there's a little bit of cleanup work in there. And then sooner or later we get to the PCR step, which stands for polymerase chain reaction, right? And we've talked about this before. So that polymerase chain reaction, or PCR, is going to take this small amount of DNA that we have and it's going to amplify it. And then in the PCR step, there's typically restriction enzymes that we will use to go in and start snipping the DNA up into the chunks. And when we start snipping the DNA into the chunks, we are really relying on the polymorphisms of the DNA from the different samples, because each DNA sample will have different areas where that particular base pair assembly is going to show up, and yours will show up in a different spot than mine will show up, and we start to get different base pair fragments that begin to happen here. So if you look on the gel electrophoresis, which is down here at the bottom, you'll see the crime scene sample has been taken, and then here is suspect number 1. Suspect number 1 has polymorphisms in whatever form that they come into, whether they're SNPs or whether they're STRs or whether they're VNTRs. So those show up, the restriction enzymes will recognize those, and they will begin to cut those in the different areas, and we start to see banding that shows up in that lane. And then here's another suspect from another person. That suspect will also have polymorphisms in their DNA. And once more, just as before, they're either going to be the SNPs, or they're going to be the STRs, or they're going to be the VNTRs. And those will show up in a different way in different locations with different base pair fragment bands that begin to happen. And that banding will look different because of that. So for particular crime scenes in this case, if this is the suspect that we, or the crime scene sample, and this is the suspect, we're seeing a perfect match with those polymorphisms or with those DNA base fragments. And because they are a perfect match, then that would assign suspect number 2 to the crime scene. Again, it does not mean that that suspect committed the crime, but it at least puts them at the crime scene, and maybe that, along with other evidence you could use to prove that that suspect did the crime, but maybe not. Maybe they were just there and was like, hey, how you doing today? And then left. Okay. All right. So if we begin to start taking a look at the lab directions for this particular lab experiment, it's going to go through this description of PCR. This is something that you're getting in our lecture videos, so I don't think that you really need to pay attention to that. And then in addition to that, though, we don't want like 1,500 base pair fragments to show up in the gel electrophoresis. If we can keep this a little bit clean, then we can compare them much easier between each other. But the more fragments that we have, the harder it is, and the more work that's going to involve in order to play match game or figure out who the mother and who the father is. So these are things that we need to maybe think about, and we need to limit the number of fragmentation cleavages that we get from these DNA samples. So here they're going to go through a discussion on PCR, and it's going to say, okay, so we're going to use PCR and we're going to analyze these polymorphic DNA regions. And now you know what that fancy word means. Polymorphic means changes. These are regions of the DNA that is changing from one person to the next, and we want to pay attention to those, because if we can isolate those and put a focus on those, we can get these different bands that begin to happen in electrophoresis. So it says, by examining several different VNTRs or STRs from the same individual, you can get what we call a DNA fingerprint for that individual, which is not like any other person. So this is something that's very unique to you, and no other person will have those exact different polymorphisms that you will have in your own DNA strand. And that's what we've always compared. That's what we've always kind of looked at in the electrophoresis gels. This is nothing new. It's not a new concept that we're introducing. We're just putting some proper terminology on it maybe now. now versus the other times that we've done it before. All right, so it says 1VNTR, which is the variable region, a little bit larger, is known as D1S80. So that's what this particular lab is going to focus on. That region, you have that region, maybe, maybe not, right? It just, your DNA is going to be different than mine. It can show up in more than one time. So that's kind of something that we're looking for here, is present on human chromosome number 1. And it says that this variable, a little bit larger region, is actually 16 nucleotides long. So that's 16 nucleotides, aka 16 base pairs. That 16 base pair sequence is repeated between 16 to 40 times. And if you're homozygous for the genotype, you will have an equal repeat numbers on both of what we call the homologs of chromosome 1, and that will give you a single PCR product when you put it into the electrophoresis. And then if you are a person that is heterozygous, then what will happen is that you will have different D1S80 repeat numbers, and you will have more than one band that shows up because of this. And in particular, you'll get 2 PCR products that can then show up on the electrophoresis gel. So really, this is just one of many polymorphisms that we can focus on in the DNA strand. And this particular one, plus maybe a couple of others, we can use for our advantage. So what you're seeing here is maybe a DNA strand here on top, and then you see a DNA strand here on the bottom. And notice this one is paternal and maternal, meaning coming from the father and coming from the mother. And what you're finding here is if you are homozygous, then the paternal one actually happens and the maternal one happens, and you're seeing a basic replica of it, right? So half of your DNA comes from the father, half of the DNA comes from the mother, and you're seeing that this particular snippet of your DNA perfectly matches, in a way, this particular snippet of DNA. So that way, when these two combine into one, you are homozygous, which means that is the one and the only one choice that you have, and therefore that one band will show up on the electrophoresis because there's no difference between the mother and the father. So that is what we call homozygous condition. So just now I also said heterozygous. Well, what does that mean? Well, again, paternal and maternal. So these are from your two parents, your mother and your father, and then maybe one parent has this piece of the DNA that looks a certain way, and then the mother has a piece of DNA that looks a different way. So these are not quite the same. They don't look the same. One is a little bit shorter than the other one, and you're seeing that this piece of the parent and this piece of the parent is going to be a little bit different. So when they come together and they form this child, we call that a heterozygous. And because they are not the same and because they don't look identical, what you'll see is two bands that show up when that electrophoresis begins to happen. All right, so here in this example, here's a gel, and that gel in number A has a DNA ladder, and that's why you see multiple bands that show up down through the lane. And then B is going to be someone that is homozygous for this genetic trait. So in that DNA sample, when it gets inserted and when it gets ran, we see one band that shows up because that band for that particular gene that we're looking for is the same in the mother and in the father both. So only one band appears. In lane C, this is from maybe a child whose father and mother are a little bit different. So when we begin to look at these, it begins to cleave. It cleaves in the right way. It cleaves at the right base pair set, but one half of the parent is looking a little bit different. One's a little bit larger, one's a little bit shorter, and that's what you saw in the example up here at the above. So what you'll see is that there's two bands that form, and the reason is because one's coming from the mother and one's coming from the father. So those are there. They're not the same length. They're not the same base pair number, which means they're different molecular weights and they travel through the gel electrophoresis in a different way, but these are polymorphisms that we are seeing in our actual DNA electrophoresis. Okay? So that's really what we're leading up to by the end of this experiment. And this is just some background information that we're giving you before this process gets started. So it says, before performing a PCR, DNA is going to have to be extracted in some form or fashion. So that can come from, again, blood, tissue, body fluid, whatever the case is. We only need a few copies of the gene, but PCR is going to really give us a number of copies. I mean, we're talking millions, if not trillions, right? Depending on how long you run them, of copies of the DNA that we can utilize to our advantage in the electrophoresis setup. So we want to kind of do this fresh. We want this DNA sample not to be exposed or just be like dried up blood somewhere. The quicker that we get it, the better that these results are going to be. So fresh DNA is always going to be better than something that's been around at a crime scene for maybe days, weeks, or months. So to make this PCR happen, in order to get to this gel electrophoresis part, we're going to have to multiply the PCR, or multiply the DNA through the use of PCR. So what do we need for that? Well, like we've discussed before, we need free deoxynucleotides, and this is what we call our nucleotides with our A, C, G, and T base pairs. So we need these base pairs so that way the replication process can use what it needs to replicate the DNA as needed. We need Taq DNA polymerase, and that DAC DNA polymerase is what's going to grab onto these base pairs and begin to assemble these things. We need primers, and the primer is going to look for the area in the DNA strand that we're really putting a focus on, that we want it to cleave and that we want it to cut at those specific locations. And we need a little bit of buffer, so we need some magnesium and some buffer in order to make this pH ideal for all of this process to happen. So like we've discussed before, again, a lot of this stuff is repeat from prior videos. We're going to have 3 stages of a PCR. So what will happen is that here we get some DNA strands. This is double-stranded, and we need to heat this DNA up at 94 degrees. So this is going to separate our DNA from each other, our strands. So we have one strand up here at the top, and we have an anti-parallel strand that runs here at the bottom. So at 94 degrees, that's when that happens. It just needs a little bit of heat, and that is it. All right, so then what will happen is that we'll cool this down. We'll cool it down to 40 to 65, depending on what our method tells us. And then that will allow our primers to go in and recognize these areas that we want to pay more attention to. So this is the base pair order, or the base pair segment that we're interested in. Whenever you find that particular order of base pairs, we want the primer to go there and we want it to locate it. So it finds it here on the top strand of the DNA. And then what will happen is that we'll warm this up to 72 degrees, so a little bit warmer than the primer attachment. And then that is when the Taq polymerase and the reaction begins to extend the DNA strand. So what you'll see is that here we get the primer. That primer recognizes the DNA strand that we're interested in. And then from that moment, it begins to copy the DNA strand using those base pairs that we have put into the mixture. The same thing happens on the other strand of DNA, because remember, you've got 2. So this 3' to 5', that's what we're seeing here. That primer is just an empty box. It's not a red one. It's just an empty, non-colored box here. That primer recognizes it on that one as well. And that primer begins to allow the Taq polymerase and the DNA base pairs to replicate that part of the DNA strand too. So what was 1 is now 2 double-stranded DNA pieces that we get to use, that we get to do reactions with, that we get to put into the gel electrophoresis. And then this whole process repeats. That is what we call one cycle. That's what you're seeing over here to the left. So the 94, cool it down to 40 to 65, and then warm it back up to 72. This is one cycle. After that cycle is complete, we repeat it. So now we heat it back up to 94, then we cool it to 40 to 65, then we go back to 72. That is our cycle 2 completion, and then our cycle 3 completion, and then we can go all the way up to 20, 30, 40 cycles, depending on how much DNA that we actually need and how much ingredient that we allow it to use to replicate the DNA with. All right. So in this particular lab experiment, they go through this abbreviation of CODIS, and this is CODIS, CODIS. And this is a combined DNA index system. And what CODIS does is it looks at human DNA, right? And we break this DNA down into what we call the chromosomes. And these chromosomes are looking at different segments of the huge, big, long DNA strand. Within these chromosomes, we are looking at certain loci. I want you to think of loci as particular locations of the DNA. So think of maybe your DNA strand as like a huge filing cabinet, and that filing cabinet is filled with genetic information. So you take the filing cabinet and you open up the doors of the filing cabinet. When you open up the doors of the filing cabinet, there's different ones, right, that you can go to. So I want you to think of those as like the drawers that you go in order to look at these particular chromosomes. Within those chromosomes, you're going to have folders, and those folders are going to be like loci that we're somewhat referring to, and those folders are going to have particular information on particular genetic traits. It is an organization process that happens here, and that's really what we're referring to. So for CODIS, what you're going to find out is that they are particular areas of the DNA that CODIS really pays attention to. And here are 13 different sites that we see within the CODIS system. And they've labeled them as A, B, C, D, E, F, G, H, I, J, K, L, and M. So these 13 particular sites of the DNA, or particular areas of the DNA, all 13 of these, think of these as individual filing folders within a filing cabinet. And when you open the folder up, you have certain information on the inside. There's 13 of them. They do more than one. And when they do more than one, that's great, because what happens here is that your 13 folders that have this genetic information in them, not everything will be the same in those folders. We all might have the same filing cabinet. We all have maybe the same folders within the drawers of the filing cabinet, but within those folders, that genetic information is going to be different. So we look at these 13 different areas, and that's what CODIS does. And it says these are the 13 areas that we're going to pay attention to. Those 13 areas will not be the same for every single person, because we've picked 13 of these that are somewhat variable different. And what you're going to find out up here in the top paragraph, it says the odds of a match at all 13 loci are less than 1 in a trillion. So if we look at these 13 areas of the DNA segment and we compare these 13 particular areas to someone else's 13 particular areas, and we look at the banding that happens in something like DNA electrophoresis or so forth, there is only 1 in a trillion chance that someone else will have the same exact DNA at these particular locations. And that's where CODIS comes into play. So what would happen is that there's a crime scene sample. We've isolated the crime scene sample. We've looked at these 13 loci, and then we've entered that into the system of CODIS. And then we extract DNA samples from different suspects. Those get analyzed and ran, and those will get compared to what we find at the crime scene. And CODIS will tell us, listen, all 13 areas of these loci match. So therefore, this probably is 1 in a trillion chance that it's going to be somebody different. It's probably that person. So that is the power of CODIS, and that's the power of the 13 loci, like the 13 Ghosts, if you've ever watched that movie. All right, so these lab directions are going to hit in the next video. We'll kind of go through, we'll talk a little bit more about that process here, and we'll walk through what we're expected to do in that lab procedure. So come back, don't leave me yet, but come back and we'll pick up from these procedure steps.